Hello and welcome to Teach Me in 10. Today we're joined by Lotte Hetkebist, who's a Senior Product Manager at Cytiva. She'll take us through the evolution of chromatography systems, including ETCA, Cytiva's pioneering platform that has helped scientists purify proteins for over 25 years. Lotte will explore the scientific and technological foundations behind the first system, the innovations driving today's workflows, and the future of protein purification. Hi, Lotte. Thanks so much for joining us today to talk about the evolution of ETCA and protein purification technologies.
Thank you, and thank you for having me.
Yeah, you're very welcome. So can we begin today by going back to the very start, before ETCA even existed, and talk about the scientific and technological developments that really paved the way for the first ETCA system in 1996?
Yes, so back in the early days, protein purification was a very slow and labor-intense work. Techniques were used like precipitation or column chromatography using gravity flow. So this meant it could take several hours or even up to days to complete a separation. Then came the HPLC systems, high performance liquid chromatography. And these included high precision pumps, and they were run with a higher flow rate, and they could also be run at higher pressure. But the conditions used were very harsh, so it was not optimal for proteins. They would denature and lose their biological function. So there was a need for a more gentler approach for the proteins. And in the Swedish Uppsala-based company, Pharmacia Fine Chemicals, which is actually the same site where I work today at Cytiva, the engineers started to develop a new chromatography system that could be run using aqueous buffers and also at lower pressures. And in 1982, the first FPLC system was launched. FPLC is fast protein liquid chromatography. And this really laid the foundation for what was to come next with the launch of the ECTA Explorer in the 90s.
Lovely. And as the field evolved in the years that followed, what were the most important advancements that changed how purification systems were both designed and used?
Well, during the 1990s, the use of recombinant techniques became more common, and it made it more easy to produce larger amounts of protein. And in the industry, the biopharmaceuticals were evolving. There was a lot of focus on monoclonal antibodies and how they could be used for immunotherapies. This led to a need for optimizing purification protocols that could later be scaled up for manufacturing of these biopharmaceuticals. And our engineers continued to work, and in 1996, the ECTA Explorer was launched, which was the first system on the ECTA platform. And this really brought modern purification into a new era. The ECTA Explorer was a very integrated system. It had a lot of valves, enabling automation and unattended runs. And the software was more modern and more easy to use. It included a lot of built-in application support, and there were ready-made method templates for the different chromatographic techniques, as well as the scouting functionality. So now there was a means to perform a lot of experiments unattended and speed up the process development work.
Lovely. And then looking at today, customer requirements must have developed substantially from the 90s. How have customer needs and requirements shaped the latest developments in protein purification systems?
Yes, so nowadays it's a lot around speed and how to get proteins purified faster. So if we look at academic research, for instance, the purification of the proteins is nowadays mostly seen as something you have to do rather than the main focus of the research. So there is a need for easy to use methods to generate results quickly, and a straightforward way to produce pure proteins to get on with the project and get to the next publication without unnecessary delays. At Cytiva, we have developed the ECTA Go system, which is an easy-to-use compact system for basic purification. And this can really be used as the workhorse in your lab. If we move to industry, process developers, they face a constant pressure to get faster to market. So for them, the question is not only how do I purify this, but it is like how do I purify this faster and in a way that can be scaled and comply to requirements. They need automation to be able to do 24/7 operations, and they also use data-driven approaches to process development. And digital tools are used to generate more data quicker and to get a larger understanding for the protein purification processes. And in the last decade, design of experiment has become a standard tool that are used among process developers. And our team, they continue to work on the second generation of ECTA systems. And with the ECTA Avant and the ECTA Pure systems, they offer a high degree of automation and also built-in operational security features, which will allow the researchers to run more experiments in a shorter time. And they can apply consistent methods, which will lead to less hands-on work and also more confidence in the results.
Lovely. And then now zooming out a little to the broader industry landscape, what are the key trends currently shaping process development and manufacturing?
Well, across both process development and manufacturing, a big trend today is how to become more efficient. How can we spend less time, work with less material, and less fixed infrastructure, while still, of course, maintaining the quality and the flexibility that is needed. So in process development, the teams are focused on moving faster and also to make better decisions earlier in the process. So this is driving a shift towards more connected workflows with fewer manual steps and methods that are designed with scale in mind or already at the start. And in manufacturing, it's a lot about how to increase the productivity without having to expand the facilities. So reducing the buffer volume used, shortening process times, and using automation to improve the consistency and flexibility.
Lovely. And of course, science itself has expanded, with scientists now working across a wider range of modalities and molecule types. What new capabilities are becoming essential in chromatography systems?
Well, during the last decades, many biopharmaceuticals have been based on antibodies or antibody variants. So for this type of molecules, we now have quite generic platform approaches available for how to perform the purification. So this will shorten the time you need to do the process development. But for the new modalities that are ranging from small molecules to really complex biologics like viruses and nanoparticles, those platforms are not really in place yet. So there is a need to collect a lot of data around the purification of these new molecules and characterize increasingly complex samples without slowing down the workflow. And this leads to an increased need for easy integration to different analytical techniques to collect all this data. The ECTA Pure system offers a highly flexible solution which can easily be tailored to fit the needs of the specific molecule that you are working with. And it's not only in terms of automation, but it's also in the possibility to connect to different detection methods.
Yeah, lovely. And looking ahead now, if we think about what's coming next, what do you see as the next major shift in how purification technologies support scientific innovation?
Well, the most burning question in science today is, of course, how to best leverage AI to improve the scientific insight. Already computational design is used together with AI to design molecules with optimized properties already when you synthesize them. And this will, of course, lead to a more predictable path going forward. And during process development, digital tools are used, like design of experiment, as I already mentioned, but also mechanistic modeling in combination with automation and smarter data analytics. And this will help to speed up the optimization and improve the reproducibility, reduce dependency on trial and error. But the real breakthrough here is not the speed that you gain, but it's actually the predictability. So when you build these new platforms and can use the digital tools, you can model purification processes also for similar molecules and thereby shortening the process development time needed. But it's also important to remember that with all this data that you need to collect to characterize the new processes, it will be very important that you keep your data organized so that you can apply the AI tools later. And another very important thing around data handling is the security aspect. As we all know, nowadays data security breaches appear all the time. So going forward, it will be very important that you make sure that your software is up to date with all the latest security patches. And with all these tools that I mentioned, computational design, AI, and in silico modeling methods, this will still not remove the need to perform actual experiments. So going forward, we will still need the chromatography systems to run experiments that can ensure that the synthetic constructs really meet the predicted specifications, and also that they perform as intended in real world. So there will be a linking between the in silico design with mechanistic modeling and experimental validation. So in this talk, I have given you a few examples on how Cytiva have been supporting purification for the last 30 plus years. As we move forward, we will continue to be your partner and support all the new ways of working that are emerging.
Lotte, thank you again for joining us. It's been a really great conversation and really fascinating to hear your insights on where purification technologies have come from and where they're headed next.
Thank you.
Thanks for watching this episode of Teach Me in 10, brought to you by Cytiva. To learn more about chromatography innovation and explore Cytiva's resources, check out the links in the description. And if you enjoyed this episode, don't forget to like and subscribe for more episodes. See you next time.